Hi all, welcome back to Anatomy and Physiology um, for everyday life. And today we're going to talk about cells and histology. So, part of anatomy and physiology, and part of really learning about the anatomy or the structures, is to know what the names of the structures, or at least what builds those structures. So, kind of the smallest living unit in life is cells. Right? And so this was discovered a long time ago by an individual named Robert Hooke. He kind of developed the microscope and was the individual to coin the term cells. And we'll talk a little more about the history. But cells build tissue, and then tissue builds organs or larger structures. And so, in order for us to talk about maybe diseases or injuries or just everyday life, giving birth, reproduction, growth, dying, just digestion, anything. Really, we kind of have to talk about cells. You have to know something about cells. And so that's what these next few lectures are going to be about. Okay. So diving in, there are quite a few different cells that humans have, like bone cells and red blood cells, white blood cells, neurons, intestinal cells, the ovum, the sperm. Okay, and we'll talk about others like epithelial cells, muscle cells. Yeah, there's lots. Okay. All right, so cells in general. First, all cells are small. And we're going to talk about why this is, okay? To some of us, it doesn't make sense that all cells would be small because if you're a really big organism, wouldn't you have bigger cells? Right? But that's not really the case. Right? Robert Hooke, as I discussed before, was the first person to describe a cell, and he did so in 1665. He was looking at cork, which is a part of, like, right underneath the bark of a tree, and some trees have more, some have less. There's an area of cells there, which is where we get cork from. So, like, if you're looking at a cork board or something like that, that's the area of the tree that that comes from. But he was looking at that, and he made a microscope, which is pretty primitive, like 30 times your eyesight is about what his microscope was, but it was good enough to be able to recognize that there's these uniform structures, and they're all kind of rectangular in shape. And he said that looks like the area where monks sleep. Okay, so monks sleep or used to sleep in these square areas, like a little teeny room that was square or rectangular. Um, and they were actually called cells. And so that was a monk's cell. It was the quarter at which they slept in. Um, and Robert Hooke said they look like monk cells. So he called them cells, and that's where the name cells came from. Okay. Now, after Hooke's discovery, then a lot of other individuals came around and they started looking at all kinds of cells. So Matthias Sladen was a plant biologist, and so he got a hold of every single plant that he could ever get and cut them up and did really thin slices of them, threw them under a microscope. Okay, so this is almost 200 years after the discovery of cells. So microscopes got better. One of the individuals that made microscopes much better was a guy named Anton van Leeuwenhoek, who made microscopes uh, for basically a living. He was very good at making glassware and bending glass to make it so the microscopes had more magnification. But at any rate, Schladen got a hold of all these plant cells. Theodore Schwann, who was an animal biologist, got a hold of all these animal tissues. And these two men didn't know each other, but Schladen discovered all plants have cells, And Schwann discovered that all animals have cells. And basically, you can see one discovered it in 38, the other one in 39, but they're doing the work simultaneously. And so they both published, and that really gave us this idea that all organisms are composed of cells. Okay. So, what are we talking about when we're talking about size of cells? Okay, so if you look at just kind of the tip of your thumb, okay, it's about 20 millimeters across. And then if you kind of did a cross section, looking at the fat layer, looking at the veins that are in there, you're looking at the muscle fibers, you're looking at the epithelial cells, all of that about 2 millimeters. If you just pull off the epithelial cells with the dead cells on the outside, or cornified cells out here, and then the living cells on the inside, it's about 0.2. So a typical cell is somewhere around 20 microns. Uh, and then if you start diving into the cell, you can start looking at the organelles. So this is a mitochondria, it's about 2 microns. If we start looking at smaller processes like how are proteins made, okay, the structures that are making proteins, maybe zero point 0.2 microns, or maybe even smaller at 20 nanometers. These are ribosomes. Okay, and then you get into things like atoms, okay, or molecules, and then an atom. And so we're going from nanometers to micrometers to millimeters. And so today we're really talking about this kind of region, the micrometer kind of area of visualization. All right, so the cell theory. Okay, again, I said Hooke discovered cells, Schleiden and Schwann came up with that all plants and all animals have cells. And so from that, scientists developed a cell theory. Okay, so theories in science are extremely well tested hypotheses. Okay, so a hypothesis is an explanation for a natural phenomena. A theory is an accepted explanation for the natural phenomena. And in that case, theories, okay, are a general understanding that this is true, this is correct, it has proof for it. Okay, it may not be proven, okay, which means that something can't be falsified, but in the case of a theory, it holds a lot of weight. It's very important to biology to have theories. One of those theories is a cell theory. And it has three tenets. Okay, the first one, all organisms are composed of one or more cells. Okay, this was Schleiden and Schwann's work. Okay. Cells are the smallest living things. This is a combination of Hooke's work, von Leeuwenhoek's work, Schleiden and Schwann's work, okay, discovering cells that, microscopic cells that can live on their own, single cells. Okay, so the cell is the smallest living thing. And cells arise only by division or by previously existing cells. Okay, this work came from a man by the name of Louis Pasteur. Okay, Louis Pasteur, which some of you guys probably have heard that name, okay, pasteurization, or the reason why we can kind of drink milk for a much longer period of time, okay, is because pasteurization was discovered by Louis Pasteur. Okay, it was a way to heat, destroy the bacteria, but not denature the proteins, and then if as long as there's no new bacteria being added or anything like that, you can drink the milk or eat the product for a much longer period of time. In addition to that, though, Louis Pasteur corrected this misconception that had been going on for thousands of years. And that was, if you get sick, it was the doing of a superior being, or a supernatural event. Okay, and lots of cultures had this ideology. It wasn't just Christianity. It was all kinds of cultures had this kind of ideology that this supernatural or something that we can't really explain, and we explained with, oh, it's a supernatural entity that's doing this, is what was making people sick. Pasteur didn't agree with this. He didn't think there was such thing as spontaneous um, events. So either spontaneous reproduction or spontaneous growth or anything like that. He thought there must be something there. Okay? And so if you leave a piece of bread out and forms on it, the explanation was that was spontaneous growth, that a superior being or higher being made the mold there. Pasteur was like, nope, that's not what's going on. I think there's something in the air. And so Pasteur did lots of different tests to test that out, and discovered that indeed that there are things in the air. There's bacteria, viruses, fungi, all kinds of things, and that all cells come from pre-existing cells. Okay, so now let's talk about these small cells. Okay, so I mentioned that all cells are small, and that's generalized that all cells are small, but for the most part, all cells are small and about the same size. Okay? And the reason for that, okay, is that large cells, the distance from the control center, okay, which is the nucleus, to the outside layer increases as you increase the surface area. Okay, so if you think of a cell, it's going to be somewhat circular or kind of have at least some shape to it. And as you increase surface area, the volume changes. Okay? But the volume changes faster than the surface area does. Okay? And so as you increase the size of cells, the volume gets bigger faster than the surface area, and therefore the control center doesn't have the ability to control that outside layer any longer. Okay, and I'll show you an example of this here in a second. Okay, because of this reason, instead of having a bunch of large cells, we have a bunch of small cells. Okay, and so bigger organisms just have more cells. Smaller organisms, less cells. Okay? It's not a matter of size of the cells aren't really different. Okay? It's just a matter of how many cells. Okay, so again, back to that cell size thing. Okay? It's called the surface to volume ratio. So as the cell increases, the volume grows more rapidly than the surface area. Okay, like I stated before, so the volume or how much space is inside is growing more rapid than the surface area. And the surface area is how much area or what accounts for the outside of that object. In this case, most of the time we're talking about somewhat circular or oblong structures. And we'll get to some other cells that are not that way. But that's really important because that outer membrane, okay, and we'll get to the different structures in case you're not sure what I'm talking about. But that outer membrane is what makes up the surface area. It's really important because it allows things to come into the cell and leave the cell. And if the volume increases faster than the surface area, then the amount of material you need to come in the cell increases as the volume does, but you don't have the surface area to provide for it. Okay, and this can be all kinds of things: water, sodium, potassium, calcium, glucose. It could be the energy sources that are coming in that you need to drive the work that's going on in the cell. You just can't supply enough energy or resources for that cell any longer. Okay, so looking at an example of this, okay, so this, it doesn't matter what the unit is. We could say this is Micrometers, it really makes no difference. But let's say you have 1 unit. Okay, that's the radius. So going from the center to the edge. Okay, it's 1 unit big. This one is 10 times the size. So going from the center to the edge, this is 10 units big. Okay. The surface area is 12.5 units. So this entire outside edge is 12.57 units. Okay. And then the 10 times bigger is 1,257 units. Okay. That's pretty much a 1-for-1. The difference is when you get to the volume. The volume of this one is 4.189 units. But then when you times or get 10 times bigger, now it's 100 times bigger than the original source. Okay. I'm sorry, 1,000 times bigger than the original source. Okay. So the volume is much larger based on that kind of surface to volume ratio. Okay. So larger cells have far less surface for each unit of volume. Okay. Hopefully that makes sense. And the reason that this is important, so if this is the nucleus, and the nucleus is, at least this region is making a lot of proteins. And let's say you need proteins out here on this cell membrane. To ship the proteins out to that cell membrane, it takes much more energy than to ship it in here because that volume has increased by 1,000 times, okay, versus 100 times increase for the surface area. Okay? So much further distance, okay, less efficient, so most cells are small. There's a caveat to that though. Some cells can be large because they buck the system. They cheat the system. So that surface to volume ratio is based on a sphere. Okay? But if you're not a sphere, but if you decide, oh, well, instead of being a sphere, I'm going to have these long projections, these long finger-like structures that hang off, okay? And with lots of folds, now you've increased your surface area far more than you increased your volume. And that's how things like neurons can get away with being much longer and thinner, okay? Because they have dendrites that hang off and increase the surface area. Other cells, like your microvilli inside your stomach lining and your intestinal tract and whatnot, okay? These cells are maybe sphere-like on one side, but the other side have these finger-like projections that reach up, and in between those fingers are lots of protein receptors and receptors that allow for nutrients to come in to those cells so you can absorb those nutrients. So that allows them to be much larger and have a much greater surface area than they do volume. Okay? And so otherwise than the few examples of just increasing your surface area, larger organisms will just have more cells. For example, humans have 30 to 40 trillion cells. Lots of cells. Okay? An elephant has 1 quadrillion cells. That's about 100 people's worth of cells. Okay? So elephant doesn't have bigger cells. If you take a red blood cell from an elephant and a red blood cell from a human, most people would never be able to tell the difference. Can't tell the difference based on size. They're very, very similar to each other. The difference comes in the number of cells. All right, so just looking at cells real quick. Okay, so cellular structure is organized, and we're going to talk about that organization, okay? And it depends on the organism, but cells can be organized based on their outside membrane, their inside membranes, based on the material that's inside the cell. So this is red blood cells. These are human red blood cells, and you can see a couple things. Okay, this outside ring is called the plasma membrane, or what's sometimes just called the cell membrane. That plasma membrane determines what can enter and exit the cell. Okay? The plasma membrane is made of phosphate heads and fatty acid tails. Okay? The phosphate heads, they're okay with being next to water, okay, or incorporated with water. Fats, though, and many of you know this, fats are what we call hydrophobic. They're afraid of water, or they don't like water, they don't mix with water. So now you have this molecule that makes up the plasma membrane where one side is good with water, the other side repels water. And so this is what keeps these things like a capsule. So you can have water on the inside and water on the outside, but they're not dissolving. They're not falling apart. The other thing is outside, there's lots of different determining factors on what's outside of the cell, and we'll talk about some of that. But inside is the cytoplasm, or sometimes called the cytosol. Right? The cytoplasm is like kind of like a jelly that's been watered down. That's what it's like. Okay? But it has water, it's got proteins, it's got lipids, it's got carbohydrates, it's got all kinds of things in that mixture that fills up the cell. And it can change over time. How plump the cell is can change, just matter of, do you have enough water or not enough water? Okay. So if you're dehydrated, those cells will tend to shrink and shrivel. And if you just drank a big old glass of water, they should plump back up.